Thanks very much for a very, very kind introduction. Okay, so yeah, obviously I'm very pleased to be here, give a seminar. What I'll do, I'll probably remove myself from the screen so you can see the slides only. All right. So I'll talk about presynaptic mechanism, synaptic function, which we've been trying to explore using optical sensors. I start with a very basic introduction. You all know that neurotransmitter release is controlled by presynaptic calcium entry, but also it is regulated by resting calcium, which is in the nanomolar domain. It might depend on the short-term history of firing. Well, historically, as you know, it's a giant synapse preparation which would provide you with basic insights into the machinery of release, something like calyceal synapses. They are large enough to allow experimental access with patch clamp and so on. But what controls release probability and the dynamic of release probability at small central synapses remain very poorly understood until now. But what has been happening recently is a technological revolution. Genetically encoded optical sensors, in particular sensors for neurotransmitters like glutamate. Okay, this is one of those papers from Luger's lab about the genetically encoded optical sensors for neurotransmitter, in particular glutamate. Right. So now we can say we're moving from the sort of historically glorious past when we had to record excitatory postsynaptic currents to understand the release of neurotransmitter to more direct glutamate imaging. And about 5 years ago, more than 5 years ago, we were among the first who tried this method when Tom, in acute hippocampal slices, expressed this glutamate sniffer, IGLU we call it, around neurons in CA3-CN1 area. Then he would patch on CA3 pyramidal cell, fill it in with the red dye, Alexa 594, and then do what few labs in the world can do, trace its axon down to the area CA1, almost a millimeter, and then zoom in. You can zoom in on an individual bouton like this, and then to scan rapidly, to monitor in a sort of a rapid fashion what happens at the bouton, we use what we call the tornado line scan, which is a spiral line scan allowing you to cover the entire area, more or less, of the bouton within a couple of milliseconds. So what happens if you image like this is what is shown here. So you've got a couple action potentials, in this case, escape action currents, triggered at the soma by this pipette. And actually what you see here is the glutamate signal as the average one. Here you've got this, degree of the spiral turns, right, against time. Otherwise, it's very similar to a regular line scan. So this is the average one. But what was important is that if you look at individual trials, you could actually see successes and failures of individual release events. All right. So that experiment showed us that we had enough sensitivity, this method, to look at the actual quantal release of glutamate from individual synapses in organized brain tissue. So then we moved to try and monitor glutamate release at multiple synapses. Well, in this case, we used organotypic hippocampal slices, express this glutamate sniffer in CA3 pyramidal cells again. Then you can trace the axon, you can sit in the cell with the pipette. And then what you see, you find several boutons of on the axon, for instance. In this case, they're conveniently located within the single focal plane, so you can see all 4 of them. And then we've rearranged our imaging routines so that you can actually image all 4 boutons simultaneously with the laser beam, jumping between the 2 and 3 and 4. So you've got all four, so the pseudo-line scans, if you like. In this case, it's without the spiral scanning, is just the point scanning, but jumping among the boutons. So all four like this. If you record it like this over several trials, you could actually observe successes and failures very easily, and then establish the release probability for the first action potential, second one, and the averages here in black line. Not only that, if you plot a histogram of the amplitude, this is what you can get. So in the yellowish one is the noise, spread of the noise around the zeros, or failures. But then what you could see, actually, how many quanta of glutamate are released. Sometimes it's very clear, nothing, 1, 2, nothing, 1, 2. Here's probably clear, nothing, 1, 2, maybe 3. We don't know. Sometimes it's less clear. But it was important to establish that this approach could, in principle, enable quantum analysis at multiple synapses in organized brain tissue again. Now, what about presynaptic calcium, which stays there at the nanomolar range, which is very difficult to measure? Why? Well, I'll try to explain you why it's difficult to organize brain tissue. So historically, ratiometric calcium dyes have been used very successfully to monitor nanomolar calcium in cultured cells. There is a problem with the brain, because cell cultures are translucent. There is not much scattering of light at all. Whereas brain tissue is an optically turbid medium. And in turbid medium, you've got scattering, which is inversely proportional to the fourth power of the wavelength. And there is plenty of nonlinear absorption going on there, which means that the green light is scattered like this, and the red light is scattered like that, which implies that the ratio between the two spectral signals, whether excitation channel or emission channel, depends, and sometimes strongly depends, on the depth in the preparation of scattering and absorption and the rest of it, which means you cannot really use these ratiometric dyes properly in order to estimate the absolute concentrations in the tissue. So therefore, we turn to the time domain and trying to use fluorescence lifetime imaging in the first instance with the green dye, Oregon Green Dapter 1. I'm not going to talk about the technique of FLIM. I guess it's sort of a technical issue, we can discuss it later. All I will mention is that we managed to calibrate in a slightly novel way normalized total photon counts, as we call it, against calcium concentration. So we've got a readout of FLIM versus calcium concentration, the absolute one. The important thing is that this measure doesn't depend on light scattering, absorption, dye concentration, laser power fluctuation, and so on, because it's a time domain. It's the lifetime. It's a lifetime of say of a candle, which could be a couple of minutes. The candle lasts, and it doesn't matter how many candles you've got, 3, 4, 1,000, whether you can see them properly or behind a curtain, still the lifetime will be approximately the same, right? That's the principle of the of the time domain. So this approach allowed us to actually image the landscapes of nanomolar calcium inside individual cells. In this case, in an acute slice. So you could see this color-coded landscape. Now, there is a challenge, however, in terms of the synaptic imaging, because the emission bands of this Oregon Green Dapter 1, which we established as a reliable tool for FLIM measurement. So emission band of this one and the the most common glutamate indicators, which are green, are chromatically inseparable, so which would preclude simultaneous monitoring of calcium and glutamate. So we were trying to find a redshifted calcium indicator, which would be also FLIM sensitive to calcium in terms of the concentration. And we tried quite a few dyes, which you could use as a synthetic dye inside the cell. And eventually we found this Cal 590. Again, I'm not going to talk about flame. This is just a recording where you got, very brief laser pulse and the different decay time of fluorescence depending on the calcium concentration. Again, we're able to calibrate this dye when you've got a flame readout versus calcium concentration, and this dye turned out to be very sensitive to absolute calcium in the range of between 20 to 100 nanomolar, which is precisely the range we want to see inside cells in terms of resting calcium anyway. So in this case, we were trying to monitor simultaneously glutamate release and the calcium dynamics at individual presynaptic terminals. Again, this is the cell in organotypic slice expressing green glutamate sniffer. But in the same cell, you could patch it and fill it in and dialyze with the red, in this case, showing us magenta here, Cal 590 calcium indicator. It looks like this. And then what you do, again, you just trace these axons and find one or two or more presynaptic boutons. And again, you use spiral scan, as I explained before, the tornado scan. But in this case, you are able to record simultaneously from two channels. One is green, one is red. Again, you've got the turns of the spiral here, you see, from the center of the spiral towards the periphery. In here, otherwise it's like a line scan, all right? The time is here. And there are 4 action potentials. You can immediately see that you've got a failure here in terms of the glutamate release and no failures in the calcium channel. And if you look at the glutamate channel only, you could see that from trial to trial, you've got failures, releases, different numbers of quanta are released as you go along and trial by trial. In this case, we are therefore able to monitor glutamate release. Here is about 20 trials, calcium signal in terms of the intensity, fluorescence intensity of calcium 590, but also actual readout of the absolute calcium concentration in the presynaptic terminal, like this one. You see, sometimes you can even spot spontaneous calcium releases to one of the trials, which is an interesting phenomenon, okay? Without action potentials at all, without glutamate release. Again, in this case, we could try and analyze the quantal content of glutamate being released. And then in this particular bouton, when we plot the amount of glutamate release versus the calcium signal and versus the estimated quantal content, you could see a positive correlation, which was very interesting because it's not trivial. So we try to look at this in more details. Before we do that, however, we have to establish whether the calcium indicator, which is calcium 590, actually buffers too much of calcium inside the presynaptic terminal, so it affects release probability. So in order to establish that, Tom ran a couple of experiments in which he patched the cell and started recording immediately, more or less, so that you've got an increased concentration of calcium indicator in the presynaptic bouton, with time, as the time goes along. So you've got from 0 to 20 minutes, this is the indicator of the calcium concentration, which is total photon count. It goes up because there is a diffusion equilibration of the dye. It takes about 30, 40 minutes normally. So you see, you can clearly see increased concentration. At the same time, if you look at the basal presynaptic calcium readout, it stays the same. And as we know, with flame, it doesn't depend on the dye concentration. You can actually read out this concentration without any bias. So it stays the same. If you look at evoked presynaptic calcium entry, so this is equilibrated calcium increment after the action potential, it stayed the same. Furthermore, if you compare the boutons in nerve cells which were or were not filled with calcium-90, you got a large scatter of release probability at individual boutons, but no difference. Right, that was very interesting. So there was no detectable effect of calcium-90 dialysis on release probability. So therefore, with them, turned to collecting multiplex imaging data from these individual synapses, trying to look at the relationship between presynaptic calcium release and presynaptic calcium and release probability. This is one of the examples. Again, the CA3 pyramidal cells. You trace its axon down there in area CA1, select one of the regions of interest, zoom in, you can find quite a few boutons there, and you don't have to sort of identify them morphologically because you could identify them by the fact that they release glutamate. All right, let's see what happens to these 4 boutons in 2 channels. The green one, again, is glutamate release. The red one is calcium, presynaptic calcium dynamics. As you can see very clearly, there are plenty of failures, release failures in the glutamate channel, no failures in the calcium channel. And that what was very interesting, that in the same axon, we've got boutons with very different release probabilities, average release probabilities, ranging from 0.5, very low, like here, bouton 3 or 4, to almost 1, like bouton 1. You can see just one couple of failures, nothing else. That was interesting. Now, what happens to the relationship between the resting calcium and evoked calcium entry and glutamate release? That method allowed us to look at this more in some details. So this graph tells you what is the relationship between the quantal content of released glutamate and resting calcium. And you can see a very strong positive correlation despite the scatter. If you look at what happens in terms of the quantal content of 4 action potentials, because this is the 4 action potentials as 50 Hertz, just to look at the short-term plasticity, again, it's a positive correlation. If you look at the quantal content versus the calcium entry, the increment, again, this is positive correlation. So although it may sound expected, actually it has never been shown before. So you've got sort of instantaneous vesicular release probability fluctuating, varying with resting calcium and evoked calcium entry from one event to the next, on a trial-to-trial basis. So that was an interesting observation. Obviously, the next question was whether the resting calcium, which might depend on the previous history of cell firing, could actually affect short-term plasticity at the synapse. Okay, this is the resting calcium. This is pair-pulse ratio, which we could measure using the glutamate channel. There is no dependence whatsoever, nothing. So it doesn't depend on the resting calcium, which was very interesting. If you look at short-term plasticity for these 4 pulses at 50 Hertz, this is the average picture of 27 boutons. So some of the boutons are facilitating, some of them are depressing, because this is the normalized response, glutamate signal. On average, they are facilitating. If you look at the short-term plasticity, however, so linear facilitation or depression, actually it turns out there is no dependence. So that was very interesting. So the fluctuation in the resting calcium, again, from trial to trial, which is slightly different from the notion of the average release probability when you've got 20, 30 trials and say, look, this is the average one. So there is distinction, just wanted to emphasize, between the average release probability and the instantaneous release probability. Obviously, they are related, but they are not directly related. Okay, so the fluctuation of the resting calcium don't seem to have any effect on the short-term plasticity. Okay, the related question, which immediately sort of prompted us to ask was how far does actually glutamate escape from the cleft? And so the question of glutamate spillover has been around for quite a while. And this method allows us, probably for the first time, to look at it more directly. So how do we evaluate the escape profile of glutamate? All right. Here I illustrate what happens actually when you image an individual bouton, which expresses glutamate sniffer and is filled with calcium indicator. This is the snapshot of the bouton. This is a schematic. What happens when we image it with the tornado line scan? Okay, the cloud is point spread function of the laser beam. This is basically the cloud of probabilities where the excitation occurs. In the focal plane XY, it looks like that. Okay, in the Z direction, this point spread function of the laser beam is actually stretched quite a bit. So what we image is a focal slab which is at least 1 micron thick. And because the boutons are not larger than 1 micron, basically what you could image is actually the projection of the bouton on the XY plane. This is what it looks like in a schematic way. You've got a tornado line scan. This is your bouton. This is what it looks like in an experiment. So what you actually can measure, if you run several trials and image the glutamate signal there and average it, you've got a heat map of glutamate like this. This is where tornado scan is, and that one, the oval, is where the bouton is. Right? So you see a hotspot of glutamate release here, and then something happens further away. So, well, first of all, we wanted to correct the geometry of our imaging because this is 3D, but we've got a 2D projection. I'm not going to be into detail. So what we used geodesic correction. It's pretty much like the map of the globe. You have to stretch it to get proper distances as opposed to the observed or visible distances here. Anyway, we've got this correction there. So we've got this data. Then we look at this data over 27, I think, synapses, 23 in this case. So what we established there were 2 types of the glutamate spreading around this bouton upon a release event. Most of them were unimodal. A couple of times they're bimodal, like this. So what we try to do, we try to normalize the decay, with distance of the glutamate signal from the hotspot, and this is the distance, and this is hotspot normalized, and this is the glutamate signal decaying. So that was very easy actually, and then we approximated it with exponential decay and got this decay constant. It's about 0.5 micron. So in our experiments, we can see the glutamate signal is predominantly unimodal. So there is one hotspot there, at least at this spatial resolution, fades away with about 0.5 micron constant. So it's probably escaping the synapse quite a distance. Also taking into account that glutamate sniffer is expressed in boutons, but is not expressed in the surrounding environment. So we're actually underestimating the escape of glutamate, something which I'll talk about a bit later. And then we ask the question whether the calcium entry and glutamate release sites are colocalized. And this is a classical question because there are several hypotheses about how synaptic physical release is controlled. Either it's controlled by the calcium channels, which are next to the vesicle, all right, or there is just overlapping calcium domains controlling synaptic release more or less in a homogeneous way. Or there is a cluster of calcium channels, in which case the release is controlled by this calcium transient, so it depends on the distance where you are. So your release probability will vary depending on this distance. So we'll try to look at this, although we didn't have enough sort of super resolution to look at this. But anyway, we'll try to, for the first time, to look at this more as directly. So we found not many boutons which would show detectable calcium entry hotspots. These are the examples. You've got the glutamate channel here, this bit of a button. This is tornado profile, heat map of the glutamate escape, hotspot there. This is calcium channel, hotspot there, and all the rest of it like this. Exactly, all right. So we could compare the hotspots of glutamate and calcium in individual butons, at least with the resolution we've got here. So we didn't see exact colocalization, which was very interesting, between the release machinery and calcium entry, if you like. So in order to estimate this quantitatively, whether colocalization is there at all or is completely random, we compared the distribution of the distances in our experiment between the glutamate hotspot and calcium hotspot. This simulated random distribution of points in exactly the same geometry. It's a simple sort of Monte Carlo experiment, very trivial. And we found that, well, there is some colocalization because the distances between glutamate and calcium hotspots were on average smaller than the distances between the randomly selected points. But it wasn't small enough to claim that there is a colocalization on a nanoscale, but at least we can say that there is some sort of colocalization on the scale of a button. So that was an interesting finding on its own. And from then, we tried to and moved to the in vivo recording and ask whether we can actually do anything like this in vivo. And we started with a slightly different project when we tried to monitor, presynaptic calcium activity and sort of astroglial morphological dynamics in the barrel cortex. I'll go through this very quickly because the rest of the slides will be the follow-up of this project, which has not been published yet. So what James did in the mouse, he would express in ventral posterior medial nucleus a green calcium dye, and then would express, using the viral expression, TD tomato, red dye, this morphological tracer, which is inside the cell, soluble protein, in the cortical astroglia. And then this is a post hoc image. So you see how it's, what it looks like. So you can clearly see individual astrocytes in the barrel cortex. And around these astrocytes, you could actually observe these butons of the barrel, of the thalamocortical axons, which are sent from the ventral posterior medial nucleus to barrel cortex. And the reason for doing this type of labeling was because we wanted to use the whisker stimulation to see what happens to these axons and then what happens to this astroglia. But this is a different project. I'll finish on that, and I'll thank Tom and Kai and Sylvain and James and Leonid and Olga for helping us to do that. And we collaborated quite closely with Jonathan Lauren and Janila Campus to get this data in terms of glutamate sniffer variants. These are my sponsors, and thank you very much for your attention.
Thank you. Let's clap for Dima. Dima, thank you, and we have time for questions. Dima, yes, I saw that Pavel Bilan wants to ask a question. Pavel, please unmute yourself.
Dima?
Yeah.
Thank you for the lecture. Actually, I follow your works, but I didn't know anything about the last part, and it was extremely interesting about release of glutamate and recording of this release from astroglia. No, actually, I have many questions, and just try me to recall if this sniffer glutamate probe is a channel or not.
No, it's not. No, it's not a channel. No, it doesn't do anything in terms of the ion fluxes or anything else. No, it's a protein which changes its fluorescence upon binding to glutamate. That's all. it's expressed on the surface. You could express it inside cells or outside, doesn't matter. It's expressed on the surface and just sitting there. It competes, when you express it in astroglia, it competes with transporters. So which is interesting thing, which is different issue we're trying to figure out how much. So actually, this escape we are seeing could be an underestimate of the.
Actually, I thought about functional consequences of this expression, and I thought that maybe it is channel, and in this case, yes, it should produce substantial changes to potential, for example, in presynaptic button. But okay, okay. Let us. I have several actually scientific questions. First of all, in the first part of your talk, you demonstrated that there are calcium gradients in dendritic tree of the neurons. And what is your opinion? What are the mechanisms which can create gradients in what we consider as a just media with free diffusion?
You're talking about this first one. Hang on a second. Just like this one?
Yeah, like this one.
Yes, we found but it was surprising. We found gradients with the characteristic length about a couple of microns, 2-3 microns gradients. They're not great, but they're about 50%. The only way the gradients could be sustained is because of their active sinks and sources, obviously, because otherwise diffusion would equilibrate very quickly. But because you've got so much buffering and sinks and sources everywhere, mitochondria, channels, the stores, whatever, there are several candidate mechanisms for how you can sustain this gradient. So you've got a gradient of the calcium buffer, a couple of sources, and immediately you see the gradient. So there are several. We don't know exactly which ones, but you could assume there are several possibilities to sustain these gradients. Now these gradients change if you looked at that paper. We showed that upon stimulation, just a stimulation without any plasticity, for sometimes the calcium concentration drops, the gradients change a little bit. So
okay, no, but it seems to me it's quite interesting because it is quite stable. As far as I remember, they can last for minutes.
Yes.
And buffers in this case cannot explain these gradients. It should be some active system. If you consider that there is a diffusion between two compartments, which are, let's say, as in this example, 5 microns away.
Sure. What I was trying to say, the immobile buffers, which there are plenty of the protein, which some proteins do not move around, and obviously others they are completely they're soluble, diffusible. So if you talk about buffer mechanisms which are immobile plus sinks and sources, that's the possibility.
Okay, and I have a question to the second part. Actually, synaptic butons are very densely located on the axon, and as far as I can see, you demonstrated that glutamate can spread several microns from the site of release. It means you have some bias in interpretation, it seems to me, because you can record glutamate increase in a certain button as a result of glutamate release in the neighboring buttons.
Well, that's true. So we're trying to say a single axon rather than single button. You're right. There must be, if two butons are close to one another by more than a couple of microns, these are further away. So if there are two neighboring butons, the fields of glutamate could overlap a bit. Yeah, that's true. So we're trying to say the axon rather than button. All right. But
Thank you. Just I want to somehow also let possibility to ask other questions. Pasha, do you have more questions?
I have, but okay, I can do it.
Okay, let me ask, and then I will give you a possibility to ask more questions. Dima, you showed that glutamate can spill over from the cleft till up to 0.5 micron in vitro model and up to or more than 2 micron in vivo. And this is just normal situation. You didn't have any pathological states, but we know that spillover can activate extrasynaptic receptors, and usually it's occurred during pathological state and lay in the base of glutamatic cytotoxicity. So do you think that under pathological condition, this glutamate spillover will be even more in terms of distance, or you stimuli somehow, I don't know, maybe mimic pathological release of glutamate?
No, we don't think that we mimic pathological thing, but there are two aspects of this question. The first one, what we image is a sort of glutamate escape, which is not the same as receptor activation. So you have to have pretty high affinity receptors, maybe like NMDA receptors, to pick up this glutamate. Not only that, the glutamate should overcome the plenty of transporters before it's picked up by the receptors. So we don't know yet what is the, I mean, we know from other studies there is some sort of slight spillover of glutamate which prompts NMDA receptor dependent crosstalk. What we show here is the actual where physically glutamate is going. We have to yet to estimate the actual concentration. Maybe it's just 2 molecules of glutamate going up to 2-3 microns. Is the fact itself that it's there, and if you've got very high affinity receptors, you can pick it up. This is important. So yes, in this paper which has just come out, we show that after the LTP induction, the spillover of glutamate is boosted actually quite a bit, mainly because of the withdrawal of astroglia. So there is, so I don't have an exact answer to your question. Definitely, it's the situation is not pathological because nothing's happening. A couple of action potentials, but we don't know from our data how much of the receptor activation is going on around the synapses. We just know where the glutamate is, so things like that.
Mm-hmm. Thank you. So please raise your hands if you have questions. If not, I will. Galina Grigorina, you want to ask? Mm-hmm. Please unmute your microphone.
I have a very small question. Thank you, Dima, for the excellent presentation. But very often in the tissue we have a so-called multiply synapses, when one presynaptic terminals doing some more than 5 or 6 or even 10 contact with the postsynaptic structure. What happened with the synaptic terminals in this case? It will be some cloud allowed synaptic terminal or not?
Yes, well, in our experiments in CA3, CA1, we don't have that many, as you know, from EM. But yes, you're right. I mean, whether it's a single synapse, single sort of postsynaptic specialization or several, there will be an escape of glutamate. If there are several release sites, the cloud, as you say, will be quite a bit larger. So So we're talking about a micron distance probably of the potentially glutamate receptors, high affinity could be activated upon the release of glutamate from a single boson. And it looks as though in vivo some reason it's probably a bit higher than in vitro, maybe because of the slice changes or whatever. So that's an interesting. We have to look seriously now at what happened at the diffuse versus wired transmission, because it seems that at least glutamate is there. So whether it sort of triggers specific receptor action and how strong it is, we don't know. But at least glutamate is around.
Thank you. But in this case, we have different astroglial cells around this synaptic, not one, and several from different sizes, you see?
If you're talking about, so if you talk about different postsynaptic neurons, then this is what I've explained. But in terms of astroglia, you've got one astrocyte always around. They don't overlap, as you know, in tissue. Individual astroglia do not overlap. So within one territory, one astrocyte, you've got tens of thousands of synapses. And around any particular synapse, there will be just one astroglia because it will be all around the place, and there will be no other astrocytes around at all. So, yeah.
Okay, thank you.
So, Pasha, please.
Dima, you have demonstrated that there is quite substantial difference in release probability between the butons. And do you have some spatial information? Are, for example, butons with high probability are colocalized, or there is no correlation between the, let's say, colocalization and actually value of release probability? And the second question, have you tried to manipulate the release probability in some way, for example, reducing LTP patterns of stimulation or LTD pattern of stimulation, and to check if there is a changes actually in release probability of particular button in particular axon?
Yeah, it's a very good question. Obvious questions, obviously. No, we haven't looked at it yet systematically. Tom tried to do it. Yeah, in the initial set of data, he couldn't see any specific sort of patterns of correlation in terms of where the butons are and versus the release probability. So we don't know. I mean, it's probably, yeah, it will take a separate study. Obviously, we are looking at LTP at the moment. And we published a small paper, or Ola Kopec published a small paper, which is about LTP using the frame scan, which has much lower resolution. We cannot see individual sort of release events, but we could see something. So that definitely changes in the pattern of probability after LTP. There is definitely an increase in glutamate spillover, and we don't know yet how exactly this happens. What we're trying to look at now is LTP in vivo. It does seem to work, so we'll figure out very soon what happens to changes in release probability in vivo. At the same time, we're trying to look at this in acute slices in terms of what happens to the population of presynaptic butons once you induce LTP, whether there is a redistribution of release probabilities, changes, and the rest of it. So I don't have a short answer. There are a few interesting things happening there, and they are more complex than just going down or going up. So it looks as though there is a change in the pattern.
Okay, thank you. And I have a question. The last part of your talk, according to your data, release of glutamate from a particular button can spread up to several micrometers, yes, from the site of release. And I would like to know your opinion, how many glial cell can sniff this released glutamate? Actually, what is the size of the area which can be affected by glutamate release in the space of glia?
Yeah, well, it's exactly what we're looking at. So we're looking at this scatter of different axonal butons, and which means the scatter of synapses in that area. And there are thalamocortical axons, there are cortical axons. Thalamocortical axons are a minority, about 10%. So we're collaborating with German guys, with Moritz Findenmeier, which just published a science paper, which, this 3D, a huge connectome reconstruction, the barrel cortex. So from that reconstruction, we know, we know the 3D pattern of synapses in the area. and we know what is the sort of more or less the density of the transporters, and we know what is the apparent sort of profile of glutamate. So we're trying to estimate what is the actual sort of strength of possible NMDA receptor activation on different distances in that area, in terms of neighboring synapses, in terms of what we see in our experiments as glutamate profile. So I think I'm sort of answering your question in terms of what we're doing. We don't have an answer yet. So we'll know very soon how many synapses, neighboring synapses in the area could be affected and to what degree based on our data.
Okay, I see. And Dima, do you have any idea about the relationship between your delta F over F and the real glutamate concentration? Because
we're running quite sort of detailed biophysical simulation experiments exactly at this moment, which we did before for various things, and we're trying to establish that, to estimate that, because we've got several constraints there. We have to put it all in the very sort of large 3D model of diffusion in the sort of torture space. We use cloud computing for that. So we're trying to estimate this. I don't have an answer. It's not that easy.
Yes, I wish you success in getting of all answers to all these questions.
Mm-hmm. Okay, if we don't have more questions, I don't see rising hands. Okay, then Dima, thank you so much for your talk, and thank you for participation. Thank you for having time to participate in our seminars. And for everybody, I just want to remind that next Friday we will have talk from Portugal by Boris Safronov, and I invite everybody to join these seminars. And Dima, thank you once again.
Well, thanks very much. Thanks for your attention.
Yeah.
Cheers. All right. So hope to see you soon.
Yeah, thank you, Dima. Mm-hmm.
Okay, bye-bye.
Bye-bye.
Bye.
Bye.